So some of you are probably too young to get this, but the part of Will's War is being played by Chris Coffey this morning. Because Will will be like, that's a little longer thing. So I'm going to step in for Will and give the introduction today for the course, just to get things started. So as most of you figured out, this is where we're at. The lifetime password, if anyone doesn't have it, is go blue. As far as the course, I'm going to start just with some introductions of the core leadership of the team has done a lot of work over the past several years pulling this together. So Will, who you met last night, I'm Chris Coffey, I'm a statistician. Lori Gutman, who is sitting back here, Robin Conwitz, who is over here, and Roger Lewis, who is in the back. So that's the executive committee. And then Sam, who basically keeps all of us in check and makes everything work, but has done a lot of work pulling this together, and we're greatly appreciative of Sam's effort. So if you have issues or things you want to discuss with the course broad, contact one of us, and we'd be glad to discuss things like that. You know, each of you are assigned to a small group. You've already had interactions with your small group leaders. There'll be more chances here in the course to interact with the team and to continue fleshing out your protocol. So I encourage you to take advantage of the time that you have here. One thing that is an opportunity for you to contact people outside of your team are in the period of time you have for faculty office hours. So there are periods of time in 3 different afternoons. So these are times where there's nothing formally scheduled, but you can have an opportunity. So these can be as broad, and they can really be anything you want. So it can be an opportunity to talk to someone in your field or related to what you propose to do for your protocol, that you can discuss your protocol. It could be an opportunity to talk to a statistician who is different from the one in your small group. It could be a chance to talk to someone who just want to get career advice. So there's really a broad range of what you can do in terms of these faculty office hours. There's a sign-up sheet or website you can use on Sign Up Genius. I would encourage you, if there are people you know you really want to talk to try to sign up right away, because it's kind of first come, first serve with respect to these sign-ups. But more details of how to do that, I'm going to turn it over to Sam to give a little bit of feedback on exactly how it will work and then how you'll pair up and you know.
So a few people have already signed up on Sign Up Genius. That's great, thank you. But for those who haven't, we wanted to just show you how quick and how easy it is to sign up. So this is the website. The links are in the agenda, page 5 of the agenda. And also in the email that I forwarded all the travel information, so you can easily connect out. So this is the Sign Up Genius page. You see that each of the faculty have their office hours in half-an-hour increments. So you just click sign up. It takes you to a page where you put in your name, your email address, and that it can send you a confirmation. And at the end of the sign up, you have an option of downloading a calendar appointment. So it's very straightforward, very easy to do, but if you have any trouble, please let me know. Shoot me an email or find me laying around somewhere. And then for the faculty, we will also send you an appointment. So the appointment will come from either myself or one of the other staff, Joy Black, or Lindsay Harris. So all of the faculty hours should start here in the pairs, and then we'll leave it up to each of the pairs, the faculty training pairs, to find a spot anywhere in the hotel. So you can go down to the second floor, there's some cubbies where you can sit together. There's also a big, long desk in the lobby as you come in, so you can go down there as well. But just, yeah, we'll start everybody off here. It's really nice outside, so we can move out to the outside as well. And there's the terrace area behind us, all of that space is out as well. Any questions about sign-up fees.
So other announcements, there's a link for evaluations for each presentation. We would encourage you, if you have your laptop or your phone, you can access it to try to fill that out in a timely manner as you can, while it's also fresh in your head. Is that we do each year, the executive, we spend a lot of time trying to pour over the content and trying to modify the course to make it as useful to you. So future cohorts of the course will appreciate your feedback you give us in terms of what is helpful, what maybe not as helpful, what you might do differently, what you think was just perfect, things like that. Other brief announcements. So in terms of the evening dinners, there is a formal dinner on Tuesday. We'll have potentially an informal period. We're still trying to sort that out. But, it'll be hard to top last year's legendary period performance. It's kind of the Monday, there's no formal dinner, but one of the things that we did last year for Monday night that is really helpful is the groups will go to dinner together. So you'll go to dinner tonight with your small group, so your leaders and the fellow students in your group. It's totally you're on your own as a group in terms of where you want to go. So just touch base in the first session is this afternoon. You guys can get a sense of who likes what kind of food or where there might be. I'm sure Sam or the other locals here can give you some suggestions for restaurants if there's a particular food type that you want. What we have found is that that's a really nice way to get the groups off on a group in charge for the rest of the week, and that it's kind of the first time that you've had a chance to interact in more of an informal environment. So you're not talking about your protocol, or you're welcome to, but no one really wants to at that point. But you can find out more, what are your hobbies, moving do you like, and so it's a good chance to get to know your fellow students and your leaders in particular in kind of a more relaxed environment. So I encourage everyone to take advantage of that. There's nothing Wednesday night, everyone is on their own. I think that's the big Any questions before we start?
Just one more thing. So, mentioning the restaurant list, we will have it at the desk, registration desk. So if you have questions about places to eat, to walk this, get that to you. And then just one other announcement about the breakout sessions. So most of the groups are on this floor, the third floor. Groups 8 and 9 will be downstairs on the second floor. If you have any trouble getting to those locations, find me, find Troy, Amy, and Lindsay in the back. Spot any of those any directions you want to go.
So I'm going to start with just kind of a general overview presentation, just kind of an overview of randomized clinical trials. A lot of this, many of you probably already know. So it's kind of you, hopefully, but I think it's kind of get everybody on the same page as we're starting the course. And this is, of the stuff that will be presented, this is probably the stuff that's not as directly useful to your protocol, but it's kind of an interesting snapshot in terms of a broad picture of clinical trials. And in particular, I try to give you some of the history of clinical trials and why a lot of the things you have to address. Is that better in the back? So as I was saying, just more of an overview of clinical trials. I can give you kind of a historical perspective on why you need to be thinking about some of the things in your protocol. One of the things that I think will become really clear as you get deeper into your protocols in the course, as many of you are coming into this because you wanted to learn more about clinical research and do more of the ideas. What we found in the course of past iterations and many of us in other experiences is many of you have a good idea of what you want to do scientifically. Doing a clinical trial, there are so many other issues that you have to address that aren't necessarily the scientific or the clinical thing. There are a lot of operational issues. There are a lot of regulatory issues that you need to be thinking about as well. And so part of this course is trying to give you the thoughts of, what is it that you also need to know or need to be cognizant of when you're pulling this together. We were talking about dinner last night, and just a good example of that is, so I work in NeuroNext, which is a network for doing phase 2 neuroscience clinical trials, where we've done a lot of protocol work. And I would say a dozen times we've set on some kind of introduction call where, there's a question where we're talking about some scientific thing and saying, okay, where are you going to get your drug for your study? The answer is, the company's going to donate it. You're like, okay, you have that set up. No, I saw my friend Joe at a conference and he said they'd be glad to donate drugs. That's really not necessarily a good sense that you're going to get drug. And if the drug is very expensive and you don't have a way in place to make sure that you can get it there, that's going to be a huge thing that you're going to have to address in your protocol. So even things like how do you get the drug, how do you get the drug to where it needs to be, a lot of these things are important operational issues that you need to think about. So I started off, I pulled this from CNN, and you can open the web almost any day and you'll see stories like this. Right, which are just things that appear in the popular media, research findings that are reported and condensed down into kind of the sound bites that you would see on a website or a TV show. All of these things come from clinical trials, right? So these come from the types of clinical trials or studies that you're talking about in your proposals. So what I want to do here, as I mentioned, is give you a historical perspective. I want to go through a little bit of terminology just to make sure we're all on the same page. Go through, again, what should be reviewed at the phases of a randomized clinical trial, talk about levels of evidence, and then some of the key elements of the protocol, which is really the main focus of what you're here for. So just to give you kind of a historical perspective, if you look at clinical trials, although not necessarily in the form that we think of them today, but you could go back historically and see a lot of examples of things that look and smell a lot like the clinical trials that we talk about today. So in the 1500s, Ambrose Paré was a doctor who was treating soldiers, and particularly this battle to capture the castle of Milan by Southern English, slaughter some foreign words, so that might be bad. But at the time, the standard treatment for treating the soldiers who were injured was boiling oil. So if you had a wound, they would pour boiling oil on your wound. That was supposed to help it heal. That was what was thought to be best at the time. Now, that's probably not the best option. But they ran out of oil. So, right, there were so many injured soldiers, they ran out of oil. So they needed to come up with an alternative. So Paré came up with this combination of egg yolks, bowl of roses, and turpentine that they placed on the wounds of the soldiers after they had run out of oil. So there's no randomization, there's nothing here. But it is a comparative type study, and that what they found was that the soldiers that had this egg yolk, oil, roses, and turpentine combination fared much better than the ones who had boiling oil poured on their wounds. And so they discovered a term never again to burn thus so cruelly the poor, right? Wounded, right? So it changed what they did. Not necessarily that this combination was better, but maybe it just wasn't nearly as bad as pouring boiling oil on an open wound. But that's kind of one of the historically first instances you see of this idea of comparative study clinical research. 1753, there was Lynn's scurvy study. That's another famous example of a type of trial where to treat scurvy on patients on British ships, it was, again, the standard thing was to give them seawater. So they give you a half pint a day of seawater to drink to treat your scurvy. So Lynn wanted to try different things because it wasn't really getting the semen back and fit in quick enough manner. So they tried a number of different things, one of which involved oranges and lemons, which we now know, with scurvy being a vitamin C deficiency is probably a good thing. And what they found was those who got that actually recovered much more quickly. One other aspect of this example, though, that I think is very relevant for today is even though they found this, and it was pretty clear evidence, the British Navy did not supply citrus to ships until almost 50 years later. So it's a good example not only of the type of comparative trials that you're talking about, but also then what happens once we find something in terms of how it gets integrated into policy. So even if there's an interesting finding, often there's a delay before it really gets into a policy or starts to affect everyday practice. One's rheumatism study. So this was a very simple example, but it was one of the first to illustrate the placebo effect. So they had several studies that were showing positive effects of treatment of rheumatic fever. This was just a simple study with no randomization in one group where they gave everybody mint water. So not necessarily something you could get approved by an IRB today. But they gave everybody mint water, and they found they got the same benefits that they got in studies where they were given active treatment. So it's kind of the first to see that there is some level of placebo effect. The first double-blind controlled trial was, in the grand scheme of things, not that long ago. It was in 1943. The British Medical Research Council Ashland trial to look at British office and factory workers who were suffering from colds. They were given Ashland versus placebo. It was not randomized in the sense we have today. It was an alternation procedure. So the first subject got Ashland, the second got placebo, and so forth. So not what we would call valid randomization, but it was an attempt to start getting at that. The first actually randomized control trial was 3 years later, again by the British Medical Research Council, looking at streptomycin at bed rest versus bed rest alone. They actually used numbers and sealed envelopes and randomization. Really, this is kind of the first example of a clinical trial in kind of the structure of how we think of them today. And it just demonstrated that streptomycin was beneficial in treating tuberculosis. So those are several examples of how we got to where we are in clinical trials. To talk about kind of more of the safety and regulatory aspects, I also think it's kind of interesting to look at some of the history. Because again, a lot of things that we take for granted today are things we got to, as with many things, because of things that went wrong somewhere along the way, right? So big changes in policy tend to happen only when something really bad happens and there's a reactionary impulse to try to fix it so that it doesn't happen again. So in the early 1900s, there was really no regulation of drugs. And you would have ads like this, that basically you could say that anything did anything, and everything that, so you had all these people going around the small towns or in carnivals selling the magic potion that was going to cure cancer, or do whatever they said it would be, and there was really no regulation. In 1906, or right before 1906, there was a death of several children due to contamination and smallpox vaccines, which led to the Food and Drug Act, which was kind of the first sense to give kind of a national regulation of drugs. But this really did not provide for drug review the way we think of it today. All it really did was it provided legal definitions for what you had to say was in the drug. And if you said, so you had to be open at this point about what was in your drug. And if you made false claims about what was in your drug, you could be punished, right, monetarily or whatever. But it really didn't require any need to show that your drug was actually safe, or that your drug did what you said it could do. So you could still say the drug did whatever you wanted it to do, as long as you were honest about what was in it, right? So keep that in mind, because that led to one of the interesting disasters, side note, I'm originally from East Tennessee, and I discovered this when I was looking into these things years ago. This took place out of a company in Bristol, Tennessee, which is not far from where I grew up. So I'm kind of fascinated with this because it's one of the few times someplace in East Tennessee actually is a famous national example. But there was sulfanilamide, which was one of the first wonder drugs used to treat streptococcal infections, right? And so it was a drug that was given as a pill. And so what many of the representatives who would go out, so you had salesmen who would go out and sell drugs, back at this time, what they found was there was a lot of demand for people, particularly because this was given to a lot of small children, but whether it could be given in a liquid form, right? Because that would be easier to give to kids, it would be a lot more ideal. And so what the company did was they went back and with their chemist said, okay, can you figure out a way to make this drug in a liquid form? And they did. What they didn't think of at the time was the solvent that they used was diethyl glycol, which is a chemical that's very similar to antifreeze. And so when they started doing this, so there was no testing, there was no, the processing GCP type stuff that you have to do that we think of today. And there were a lot of people that died as a result of getting this. And this is one of the first efforts by the FDA to try to recall a drug. Interestingly, given the legislation that was in place at the time, the Food and Drug Act, it was not a crime to have a drug on the market that was unsafe, right? There was no safety review. It was only a crime to say that the drug had something different in it than what you claimed it had in it. So again, this is kind of like the Al Capone type issue. The FDA was able to pull the drug not because it was unsafe, because the type of alcohol that they said was in the drug was not technically the same kind that was actually in the drug. So they found kind of a loophole to blow from that. But even given this, 100 people died, they were not able to pull the drug because of safety reasons. They had a huge, there's a great book about this, about how they had to go through and pull this. But that led to the Food, Drug, and Cosmetics Act, right? Because here you have this big disaster, and there's no way to pull things because of the safety concerns. So in 1938, the Food, Drug, and Cosmetic Act was the first set of laws that gave the FDA the right to oversee safety of the drugs. What this did, though, was it prohibited the false therapeutic claims. You didn't have to necessarily show that it was efficacious, but you couldn't claim that it did something that you hadn't shown. And it allowed the FDA to block or delay marketing. But again, there was really no approval process. So the way this worked was that you had to submit to FDA, but if you didn't hear from them in 60 days, the default was it was okay, right? So it was more like innocent until proven guilty, unless they said you can't do this, you were fine to go. So it was a little better than what was there before, but there were still problems. And then there was thalidomide, which many of you are familiar with, which happened where you had a popular sleeping drug. It was never technically approved for sale in the U.S., but it was given because there wasn't really legislation that would prohibit giving that, and you had a lot of bids for defense. So as a result of that, that led to the Kefauver-Harris Amendment in 1962, which is really what led us to more of what we think about from a regulatory standpoint today. So there was a proof of efficacy requirement. The default of approval was removed, so now you have to get approval before you can market it. And then similar to something that may affect several of you in your protocols, depending upon what you're working with, was this investigational new drug application, which is something that you have to file in order to get an exemption to use an investigational drug to give to patients or subjects in a clinical trial that you're working on. Because technically, it's illegal to give a drug that has not been approved in the United States to a patient. If you're doing it for research purposes and you have approval through an IND, that can give you the right to give an investigational drug to a patient in a trial for the purposes of that trial. So a lot of these things are things that take place, but you need to be very cognizant of that as you pull your protocols together, particularly if it's a drug where you would have to have an IND and the regulations that go along with it. There's a lot of things about this, but I think it's interesting to know the history because it's really relevant to a lot of discussions that are going on today. So the world has changed in drug development and clinical trials in general, and many in a good way. So there are much more collaborative relationships between researchers and patient organizations. Patient advocacy groups are much more involved now through the internet, through social media. It's easier for patient groups to connect. And so in a lot of, particularly in rare diseases or diseases where there are no cures, there's a lot of pressure where there's kind of a push-pull between, so the regulatory environment, which says a drug needs to be shown safe and effective before I can give it to a large number of people. And as a researcher, I get that. From the patient side, if you have a disease where there are no options, in some ways you don't care if it's been shown that it works. If it's safe, I want to get access to it, right? So there's kind of this push-pull between those two frameworks that is still very relevant in the news today and still comes up a lot in congressional discussions, patient groups who want to do that. So I think it's important to know how we got there, and what are some of the bad things. But I do think those are the type of discussions that will come up very much. Areas like ALS, which is one where there are not good options in there, if you've read about the pressures there, in terms of what's the Duchenne, yeah, it's still my mind. Duchenne, which has been in the news, right, a lot with the drug where it's very controversial, where you have a drug that kind of looks safe. From a pure sense of what I know and what I've read, it's not clear that you could say it's been proven effective, but it's very much one of those that's kind of right on that threshold of whether does it make sense to hold it back from patients to have a high level that's efficacious, or should you start to give it? There are pros and cons to both ways, and there's probably not a right answer. But it's really something that as you develop your careers, it's going to be important things that will happen that will affect the studies that you'll do. I pulled this up as kind of other key dates in the history of clinical trials. So there are other things, the Belmont report, things that come out from informed consent and some of the things that that came out of World War II and some of the things that were done. We talk about Nazis, but there were things in the U.S. that we did that we're not proud of. Tuskegee is a great example where, we were in some ways just as much involved as some of the things we pointed at other countries. The Orphan Drug Act, which is important, there are differences if you're doing development for orphan drug that have some advantages to try to encourage companies to develop meds for orphan drugs because there's obviously not as big a market for that. So you're not going to recoup as much on your investment for a drug in a rare disease and a disease where there's a huge pool of patients to do that. So just to kind of shift gears a little bit in terms of the terminology. So for clinical trial, it's just the simplest as an experiment testing medical treatments or prevention strategy. Most of you already know this. It's prospective, so it's not a case control study. You're enrolling patients, following them over time, and it's comparative. A randomized clinical trial is one that has at least one control treatment and one test treatment. And there's some key things to think about. There's blinding or masking. I'll say blinding most of the time. Work with the National Eye Institute, masking is the more politically correct term. But there's single blind, which means that the patient doesn't know what they're getting, but the researcher does, and there's double blind, right? So normally we strive for double blind experiments. We have a lot of discussion when we're designing trials of what's the purpose of blinding, and a lot of these things are not necessarily to roll out bias that's easy to define. A lot of it is just subjective or unconscious bias. That if someone thinks they know what is best for their patient, or does that influence their actions if they know what they're actually getting? So double blind, where there's no way that would interfere with things, is the cleanest. If you can't blind, so if it's a surgical intervention or there are other ways where it's just not feasible to blind, then it's important if you can't blind the intervention to think about blinding the outcome assessment, right? So maybe if it's say some cognitive outcome that you're looking at, you have a blinded rater who would interview the patient who doesn't know what they got, even though the investigator might. So that's a way to try to, basically what you want to do is to try to keep your endpoint, the assessment of your endpoint, as independent as possible from knowledge of what they actually got, so that you avoid any bias, whether it's on purpose, which is seldom the case, or subconscious bias, which is much more potentially relevant and much more feasible to creep into these types of studies. There are different types of treatment, different types of control. So drug, that's the most common that you might see, and that's probably what I would default to saying. I'm talking to many others here as well, but there are other types of treatments. Some of you may be thinking of other types of treatments in your protocol, whether it's a surgical procedure, a medical device, therapeutic modality, diet, behavior, exercise, really any type of intervention that you think there's benefit, that could be a potential type of treatment in your clinical trial. Similarly for controls, we often think about placebo. It could also be another drug regimen, so an active control, right? If you're working in a disease area where there's an approved treatment, it might be unethical to randomize patients to placebo because you'd be withholding an active treatment. So maybe you need to show that it's better or it's not worse than the current procedure. Adding an additional agent to a standard regimen, so you see this in cancer a lot, where you have single agent versus combination therapies, to see whether there's a benefit of that, or it might be you're looking at different doses. So you can have the type of treatment, the type of control, there's not one answer to that. A lot of it will depend on what is the question that you're looking at, and what makes sense that you need to address in your study to answer that question. In terms of the phases of clinical trials, so phase 1, these are the earliest. These are mostly the first in human. So a lot of this is in healthy controls where you're just really seeing how high can you go with the drug. These are the type of things, if you walked around campus, there might be flyers ready to make $500 over the weekend, come in, you're going to be given this experimental compound, they're going to draw your blood once an hour, for a couple of days or however frequent to see what happens, how high can they go before bad things happen. So this is, as much as anything, human guinea pigs, right? You're asking people who have very little chance of we have very little chance of doing this, and sometimes things can go wrong there. Classic example is Viagra. So Viagra was originally developed as a blood pressure drug in some of the early phase studies. It was given to young, healthy male individuals at doses that are slightly higher than we recommended for a male in their 50s or 60s who are taking it today, and they had some pretty severe side effects. When it wasn't found to those side effects led to a whole different line of treatment for that drug. It led to a lot of money for the company that did that. So, a lot of times the bad things that happen, you can learn from them, and it might shift gears there. But it's the type of thing where the goal of these is often to see how high can you go in a healthy population before you go into the disease population. Because obviously if you treat a certain dose and have problems in a healthy population, you're probably going to in the disease. They're also phase 1 where you're treating the disease population to try to see how high can you go. And there's generally an assumption, although not always true in these types of studies, that the higher the dose, the greater the safety concerns and the greater the efficacy. That's not always true. Sometimes there's a leveling off of efficacy, but that's generally the assumption that's made. Once you kind of have a sense of dose, or at least a broad sense of the dose range, you move into more of a phase 2, which is the proof of concept phase. So here, you're looking at additional safety, but you're also trying to get a sense of the go/no-go decision. One of the things I will tell you in your phase 1 and your phase 2, or even phase 3, is never in your protocol, and many of you may have this language in your protocol, but I'm not picking on anyone, but it's a pet peeve of mine, is don't say you're going to show that the drug is safe in your protocol. You're never going to show that your drug is safe, right? The drug that's approved, right? We have post-approval marketing to make sure the drug is safe. All you're doing in your protocol is you're ruling out a level of adverse risk that would prevent moving forward, right? So if you say that safety is a primary aim, you're not going to show that the drug is safe. If you come to me as a statistician and say, what sample size do I need to have to show that my drug is safe, I'm probably going to laugh at you, right? Because you would need an infinite sample size to show that. The question becomes more, okay, how much risk are you willing to live with, based on your phase 2 study before you would move to phase 3? And then you can determine that, right? If you say, well, if this increases the risk 10% above what I see for placebo or some type of primary adverse event, that you can put in place. That doesn't say the drug is safe. It just says the drug is not that, it doesn't have that level of a risk. And that's really all we can say in terms of moving these forward. So that's an important component of the phase 2 studies. The other is the proof of concept. And that's more, is there enough evidence of efficacy here to justify moving forward to the big study? And it gets a lot, so the discussion last night had to do with a lot of the challenge in designing these types of studies is designing them in a way that you can weed out things that don't work before you get into the large, expensive phase 3 clinical trials. Okay? What often happens is someone is so convinced that it works that the phase 2 is set up where it's going to go forward almost regardless. I've seen many situations where there's really no question in the phase 2. It's just to get data to plan the phase 3 study. So you've already made up your mind it's going to go forward. Even if the drug doesn't work, you're not going to weed it out in phase 2. That's not really what you want to think about in setting this up. You want to set it up so that there's some minimum threshold that has to be met. So you can give it some type of rigor to answer that question that has to meet that to meet your go decision, right, to move forward into the phase 3. If it doesn't, then perhaps it's something that should go forward in phase 3. Maybe you have a drug that doesn't actually work, and it's much better to learn that in phase 2 than it is in phase 3. That being said, these are very difficult to design, as many of you are probably going to find out over the next few days. In an ideal world, they're difficult to design because you can't design them to show significant differences on the endpoint that you would use in phase 3, because you're going to run into one of two things. The two ways that I've seen that done is, I know it's underpowered, I just want to do it to get data. Well, that's just an underpowered phase 3 study. Again, what I mentioned before, it doesn't answer any question of interest, so you're going to go forward regardless, so it's almost a waste of time and resources to do the thing. The other is, I want to power it, but I want to power it for about twice the effect that would actually be of interest in the phase 3 study. That's almost just as bad, because you might, if you don't get a significant difference, but you say, well, it's still something that's meaningful, you're going to go forward anyway. So both of those are situations where it's almost like you've already decided you're going to go forward. You're just setting up the machinery of a phase 2 trial to make yourself feel better about the process, but there's really nothing that you're going to leave out. In an ideal world, what we would like to have are biomarkers that we could power on in phase 2. So that you have a biomarker that's related to the path of what you think you're going to intervene on, but you can measure differences much shorter time frame than for the clinical, and so you have to meet the threshold for the biomarker in phase 2 to move forward to phase 3. Unfortunately, particularly in the neurosciences, there aren't a lot of good biomarkers currently that exist that are validated to the point that they would serve that purpose for research. So that's something that's sorely needed, and something that a lot of people in different areas, again, as you heard last night, are looking at. How can you get better biomarkers, and that's kind of what we need. But until we have those, we're still left with this kind of challenge in how to design phase 2 proof of concepts. You'll hear talks later in the course by several statisticians on different designs that you can use. For instance, the utility design is one to try to address this question as best we can with the current knowledge that we have. But it's something that you need to carefully consider when you're putting this together. Particularly if you're, and proof of concept studies are also something that is very different in terms of how they're approached between industry studies and NIH-funded trials. So with industry studies, they're often put together and the rigor of a single study may not be quite as high because it's part of this whole program. And you're always thinking of the program as a whole when you're designing a study in industry. In more academic trials or NIH-funded trials, we really don't think of that way, and they're not really assessed that way because we're going to go to study section who's looking at the properties of that trial, not necessarily how that trial fits into the big framework. They might put that in, but it's not considered in the same way. So the level of rigor of an individual study is really assessed very differently in those two different frameworks. So depending on where you're going with your protocol, that's something that you will definitely need to keep in mind. Phase 3 doesn't require a lot of discussion. That's what we think about as the standard, confirmatory. Typically, to get the approval, you need to have 2 adequate and well-controlled, phase 3 type studies. This is where you're randomizing. You're trying to get clear evidence of efficacy. But mostly, what we're doing is the superiority studies. There's equality equivalent studies. There's non-inferiority, which you'll hear a little bit about here. Do you guys know what non-inferiority studies are? Right? Where you're basically, if you have an approved treatment and you can show that your drug is no worse, but perhaps it has better side effect profile, that could be sufficient to get it approved. Phase 4 are more the long-term surveillance studies. So these are really post-approval, post-marketing. So there's still, as I mentioned, safety. You won't show that the drug is safe. You continue to collect evidence to make sure that there's no, maybe you did a large phase 3 study in 4,000 patients and got the drug approved. If it turns out that there's some real deadly side effect that occurs in 1 in 10,000 people who take the drug, but then once it's approved, that huge, millions of people take it, you're going to start to see that. It would have been really difficult to see that in a phase 3 types of study. The other things to take into account are there are often a lot of differences in terms of populations who are enrolled in the trials and who might take the drug once it's approved. A great example of that is, so I did some research in the area of weight loss years ago, and back, again, many of you probably don't remember this, all my examples are getting dated, which probably means I'm getting dated. Ephedra was a big discussion back in the late 90s, which was a weight loss drug. And there was a lot of discussion and legislation about that of whether trials had shown that ephedra was safe and effective, or whether it was, potentially a problematic drug. So a lot of discussion there with cardiac risk associated with the drug. And you kind of look at that, both were true. You might ask, how can both be true? In the clinical trials that were done for ephedra, there were a number that were done around that time. To get into the trial, you had to have a need for weight loss, and you couldn't be enrolled if you were at high risk of cardiac events, right? So there was kind of a cardiac screen. Those people would go into the trial. It was shown that for those people it was safe and it was effective, and that they lost, a decent amount of weight versus those who didn't take it. Once it's approved, think about who would take a weight loss drug. Now you don't have that screen. So you're going to have a lot of people who are at risk of cardiac events taking it because they want to lose weight, and you see a lot of cardiac events. So it's a kind of a nice example where even though what we learn from clinical trials doesn't tell us everything about what's going to happen to the drug once it becomes approved and used by the general public, if the population who might take the drug differs in some way from who is in the trial. That's a really extreme example, but it's not that different from a lot of other situations, because there's a lot of push-pull when you're building the inclusion-exclusion criteria together from a clinical trial in terms of generalizability versus trying to keep the results as clean as possible when you're trying to assess whether it works in the population. There are also comparative effectiveness trials, which don't necessarily factor into the protocols that you're working on, but are something you're going to hear a lot about as you develop your careers as clinical researchers. This is something that, with the Healthcare Act several years ago, has gotten more emphasis in that it is looking at really from, this gets more into the policy type discussions, that if you have multiple drugs approved for the same indication, can you get evidence on which one would be the best to start on? And you also get into the personalized medicine. Are there different characteristics of individuals where if this person has this characteristic, they should be started on drug A first, this person might have a different set of characteristics and be on drug B. Historically, there's been little done in that field, even though it makes a lot of sense to compare that, mainly because if 4 companies have the drug approved, it's not necessarily in their best interest to fund the drug, comparing them to see which is best, because there might be 1 winner and 3 losers that would come into the market share. But there's more emphasis through government funds to try to do that and to try to get a better sense of where that would be for things like treating hypertension, treating diabetes. What drugs would be optimal? How can you use insurance funds most efficiently in terms of treating some of these types of diseases? So you'll hear more about that. That's a field that just continues to increase in terms of interest and discussions that you'll hear even over the last several years. It continues to grow. There's this idea of level of evidence. Many of you are familiar with that. I won't talk about that as much of it today. You'll hear like level 1 means that there's randomized clinical trial evidence. Level 2 is more that there's epidemiological studies supporting that, but maybe not the randomized trials. Level 3, which is really just more consensus or general opinion. But when you hear results, and this affects clinical practice, is this is a recommendation, but what is the level of evidence supporting that recommendation? And that's important to keep in mind what that means. If there's a recommendation that has level 1 evidence, that means it's been supported by randomized clinical trials that have been done in a very controlled environment. When it's level 2 or level 3, it's typically not as strong. Unfortunately, the general public doesn't understand that characteristics. There's always to them conflicting evidence as studies come out when you're all time today. Things are good for you, eggs are bad for you. Things are good for you, eggs are bad for you. If you look over years, that has changed constantly to where then people become skeptic of anything you say. So just the last thing I want to talk about are key elements of the protocol. So I've mentioned many times, and particularly in the proof of concept type study, that every study, and this is one of the key things that you probably already started to address in your protocol, will be important to take up, is what is the key question, right? Every clinical trial should have a key question, and that question should drive everything in the study, right? Because that's going to determine what your primary hypothesis is, which should determine what your analysis will be, and it will determine what your sample size will be, and everything else is going to fall into that. And that's what you're seeking to answer at the end of the day. And it's important that you clarify the question, because then you can flesh out all the aspects of the trial to answer that question, right? So one of the things that is really problematic is when the trial can't answer the question. So you have studies that are positive or negative, and saying that something is you'll often hear if a study shows statistically significant results, it's positive. If it doesn't show statistically significant results, it's negative. And I don't necessarily like that terminology, because a study that shows a non-statistical result, as long as it was powered, adequately powered, that's a positive result, right? If you have ruled out benefit of an intervention that there was some level of support to think that it worked, that's a win, right? Because you've shown that it doesn't work. The big loss there, and the one that is very problematic, is when you get a non-statistically significant result in a situation where it wasn't powered. Because then you don't know, is the non-significant result because the intervention actually didn't work, or because the intervention worked, but you weren't adequately powered at the level you showed that it worked. And then you've wasted a lot of time and resources to do a trial that really gives you no more information about the question than you started off with. You really haven't answered the question you set out to do, and you're kind of left with an intervention that may or may not work, you don't know. Or, most harmful is you do that, the study is incorrectly perceived as negative. Let's say you did an underpowered study. You get a non-statistically significant result. People assume, okay, that's negative, this drug doesn't work, that drug goes on the shelf and is not looked at again. If that was an effective drug, you probably just killed that line of research for years, unless somebody picks that back up. That's a big problem. So when you're answering that question, it's important to make sure that you have set up your design in such a way to answer that. That's another downstream implication of setting up studies where there's really no clear rigor. You're just kind of doing it and pushing things through and going to go forward. So I would encourage you to spend a lot of time thinking of what's the clinical question. And this is where you'll probably need to talk to the statistician in your team a lot, as you've probably already done. So one of the big aspects of getting the question clarified is you guys probably have already figured this out. The statisticians could get different language than things, right? And so often you have your question well defined in clinical terms, and the statistician on your team will be the interpreter who will try to take that and put it into a workable hypothesis that can then be put in, a sample size calculation, put the details on it that you need in order to give it, the rigor to put a testable statistical hypothesis test on and the sample size. So there's a lot of back and forth that you need to kind of speak that same language. And one of the things you can really tell, when you're looking at a protocol or looking at a grant application, is when that has happened or hasn't. Because when that has happened, everything flows well together. When that hasn't happened, it's like there's a chunk of clinical text pasted here and a chunk of statistical text pasted there, and you can tell that there's really no integration, right? So it's important to develop those relationships, not just with your teams here, but as you develop your careers and you work with statisticians at your home institution or wherever it is, make sure that you have that discussion going so that all of that is well integrated into your protocols. There's the primary and secondary objectives. The primary objective obviously hinges very importantly from your key question. So the primary objective of the study should be the objective that answers the key question that you set out to do in your protocol. But there's often a number of secondary objectives. One of the things you have to be very careful about, though, is there's a lot of interesting things that you can collect and ask in a clinical trial. And there's a lot of desire to do so, particularly, we're spending millions of dollars to do this clinical trial. Let's learn as much as possible. But you have to also weigh that against if the secondary objectives are very expensive or very time consuming, and you end up spending more time to address the data or collect the data or deal with problems with the secondary objectives, and that pulls away from assessing the primary objectives, that's a bad thing, right? So the primary should always be your major focus. And secondary should come along as long as there's a good scientific basis for them and as long as they don't interfere with the collection of the primary endpoint. Yeah, right. A good example of that, if you have a secondary objective that might be hard to collect, or say it's some type of test that's uncomfortable for patients, and somebody might go through that test once and say, I hated that test, I never want to do it again, I'm dropping out of your trial. That's a bad thing because you're losing data on your primary endpoint as a result of something that you didn't necessarily have to have in your study to answer the primary endpoint. So as you think about things you want to add, think very carefully about what they contribute to the study or what they could potentially contribute in a detrimental factor in terms of your primary endpoint. And this gets to the point I made in terms of taking it from clinical language in terms of something that's testable. So an example, for a primary endpoint, let's say you're doing a study in stroke, and you say, I want to show a decrease in stroke rate. Right? There's a lot of different ways you could show a decrease in stroke rate. Right? So you really have to be a little more specific in terms of thinking exactly what you want to show. So you might say that, to put that as an endpoint, that'd be the reduction in the proportion of patients, with a stroke over 3 years, right? So it's what's the time period, what exactly are you trying to show? Or if it's PD functional, you're doing a Parkinson's disease study, I want a slow functional status. Again, there are a number of different ways you can do that. So you have to give it a little more detail at the change in the rate of decline, and the functional status is measured by the mean change in MPS-UPDRS over a 1 year period. Right? So that you really just need more level of detail. The other thing that I gave is when you see a grant or a protocol that says study has 90% power, right? That means nothing. Every study has 90% power for something, right? You often, in your protocol, you want to give the level of detail. What assumptions did you do, right? It has 90% power based on some level of assumptions that you made for that endpoint. And you need to know the assumptions to know whether that sample size calculation makes sense. So all of this is really thinking carefully about not just the broad sense of what you want to do, but the level of detail of how you're going to do it. We talked a little bit about the inclusion and exclusion criteria. There's often kind of a push and pull there. You'll get into that in the discussions. The study protocol, that's really the key thing of why you're here in terms of trying to flesh that out. I pulled up here, this is from areas that are in the outline for the NeuroNext protocols, which was extracted from the sample protocols that are on the NINDS website. So these are kind of the general areas. For a given study, there might be different things that you need to add. But in general, the type of things that you would want to address in your protocol is study objectives, right? Make sure you're clearly laying out, like the primary objective, which gets to my earlier point about what is the question that you want to look at. What's the background for the study? What's the rationale? What's the supporting data? What's your study design, right? Selection and enrollment of subjects. What's the study medication, drug, or device? This gets into not just what it is, how you're going to give the drug to patients, how you're going to measure adherence. So that's another important thing to think about, is often we like to have some type of sense at the end of the study of if people actually take the drug they were assigned to get a sense of how adherent people were to the drug. Any clinical and laboratory evaluations, the management of adverse experiences, so that's an important thing to keep in mind. Do you need a data safety monitoring board? Do you need an independent medical monitor? How are you going to set that up? Who's going to review your serious adverse events? Depending upon the level and complexity of the study, some of the studies can be handled with the PI doing that. It's often cleaner if the PI is not the one that's reviewing the serious adverse events and making a decision on how severe they are, because you're naturally in a conflict of interest, right? If there's something that really bad happens that would cause your study to be shut down, it's a little different when you're the one making that determination. It is your study versus somebody who's independent of the study looking at that data. The criteria for intervention discontinuation. One of the things that always comes up in our next protocols that we work on is if somebody wants to come off of drug, can you keep them in the study? So there's a concept known as intent to treat. You guys are familiar with intent to treat? Right, so under intent to treat, everybody who's randomized needs to be accounted for in the analysis. So if somebody drops out, you have to do something with that data under an intent to treat analysis. So your missing data plan is going to have to be modified. If you can keep them in, even knowing what happens off drug is better than having no information on them. So if you can keep people in the study off drug, that's something to consider. That's operational complexity, but it's something to keep in mind from an analysis standpoint. There are statistical considerations. Many of those we've talked about: the design issues, the outcomes, the sample size, the data analysis, the data collection, monitoring, adverse event reporting. So what are you going to keep? What type of monitoring? What type of quality assurance are you going to do? The human subjects. Publication. So it's kind of important when it's particularly when it's a multidisciplinary clinical trial to sort out a publication policy early on. Because with clinical trials, a lot of people put a lot of effort in that, and the publications won't come till the end. You can really end up with hurt feelings if you haven't sorted out who's going to be on the primary writing teams, who's going to be lead author, who's going to be second author. Those are the type of things that you would want to sort out as early as possible. So that's a very broad overview to try to set the stage for what a lot of the other discussions are going to talk about. I think we have 5 minutes or so if there are any questions. Yeah.
What are the things for drug trials? Can you do the same for device?
They're roughly the same, although the objectives are slightly different in device trials. So usually in like the early phase device trials, you need to show that the device does what it was intended to do. So like the proof of concept aspect of the device trial is slightly different from a drug. But the general overall principles of what's the question, how you're setting up, are going to be the same.
So a study protocol is pretty long often, and the 12 pages that you have for an NIH proposal to be short. So do you have any thoughts on kind of what you take from a full protocol and put into that 12-page proposal? And then I've never submitted an R01 for a trial before, so is that full protocol something that you actually include as an appendix, or how is that handled?
You typically do. The reviewers, I think it's right for journals, you don't need to do for the typical things. The thing with the appendices, though, are the reviewers are not required to read all the appendices, so you still want to get all the key detail into those 12 pages. I wish I had the magic bullet for how to do that. It's very difficult to take a full protocol for a clinical trial and condense it into 12 pages. I think the things that you really have to think about are to hit the key things. So what's the scientific rationale? What's the supporting evidence, right? Particularly what's the preclinical evidence that supports it? What you propose to do, and then also the statistical aspect. So of those, what I tell people that work with me is the statistics should probably be at least 2 to 3 pages there. Because every clinical trial, almost every institute, you're going to go to a statistical reviewer, and they're going to really want to know that level of detail. One of the things I think is a mistake I see people make is they want to just shove the statistics at the end as a very small section. So I think you need that. I think you need the preclinical. I think what people tend to do probably too much, you don't need this, a lot of details of the operational aspects of the trial. You don't need as much of that in the 12 page. You want the scientific rationale, you want the statistical aspects. The piece that's not in there, and this is another operational piece that you guys will need to think about, because it's probably something that's the least appreciated aspect of clinical trials, data management, right? How are you going to get the data in? How are you going to design your forms? So there's a saying you learned early on, statistics, garbage in, garbage out. If you collect your data poorly or in a way that you can't make sense of it, the statistics aren't going to help. So you would want to kind of address that. How's the data going to flow? How's it going to be clean? So kind of those high level areas, not so much the specifics, that can be in your protocol or your manual of operations that you might have as well. That's kind of a vague answer because I'm not sure there's a right way to do that. Writing grants are, it's interesting, writing grants are a scientific exercise, but it's really more of an art form than a science. Other questions?